Hi, welcome to week 2 of Bio 201, Cell Bio at Northwest Indian College. This week we will review water and carbon reactions from last week, and we'll start moving into protein structure and function. Last Thursday we went over the properties of water that make it sustain life on Earth. This included that water is a polar molecule, that water is able to stabilize its temperature, so it has a high specific heat, that water is an excellent solvent, and that water is cohesive. Something that I wanted to show you in class today was a video that shows the three-dimensional structure of water molecules when they are frozen. They have a crystal lattice that is shown in this video. If you click on the video, what I want you to focus on is determining what are the red spheres and what are the white spheres. And I would also like you to think about, while watching this video, how much space is between the water molecules when they're in this crystalline form and how that affects the density. So once this space is formed when water freezes, it becomes less dense than water. So ice floats at the top of water. For a little review on these topics, I found some helpful videos that are entertaining. They're also nice to watch as well as educational. The first one is about why does ice float. I've included how long the videos are, so this video is about 4 minutes long. The second video I've included is describing functional groups, and it's about 4 minutes and 42 seconds long. And the third video is cells and the origin of life on Earth, which is 7 minutes and 29 seconds long. In the next few slides, I will talk about functional groups. It'll be, well, in the next slides, I'll explain what's going to be important for you as students in this class to know about functional groups. The video about cells and the origin of life on Earth, this will play into the lab this week. So sit back, relax, and enjoy some nice science movies. Linking carbon atoms together. So these next few slides will be about functional groups. Those functional groups are carbon molecules that make up the elements of life. And if you recall from lecture, the gas in the Earth's atmosphere over 3.8 billion years ago was comprised of nitrogen, ammonia, and carbon dioxide. We also covered that 3.8 billion years ago, these gases began the process of the generation of simple molecules, which is what I'm referring to as the functional groups. For review of this process, look in the book. There's a figure in your textbook that you can look at that shows how there's 3 different images. There's an image of the ocean with the gas interacting with water, and then over time you get the generation of the more simple molecules of carbon-carbon with carbon-carbon bonding. And this was something that occurred because with energy and time, the simple molecules started to generate more complex organic molecules, and those included the functional groups. I've included here an image of some of the energy processes that might have helped generate the carbon, those simple molecules on Earth's atmosphere, and there's a link to where this image came from. So what am I talking about when I refer to functional groups? Here are 6 functional groups that are commonly talked about. That includes so the table shows the functional group name. It shows the ball and stick structure of the carbon of the functional group. And then the third column are the properties. What I want you to focus on from this table are the name of the functional group and the properties of that functional group. So a hydroxyl is a carbon group. So R, when you're looking at these structures, means that there's carbon bonds on this end of the molecule. So if R is here, so R means that there's carbon-carbon bonds on this end of the molecule, and they're attached to an oxygen, which are then attached to a hydrogen. So a hydroxyl functional group has an O and an H, and it's a polar molecule. A methyl is a functional group, and R shown here is that there's a string of carbon-carbon bonds on this end of the molecule, and on the other there's this tail that has a carbon and 3 hydrogens. And this is a methyl group, and this is the nonpolar. This is a nonpolar molecule. A carbonyl is a carbon attached to 2 other carbon functional groups. These R's represent other types of functional groups or other carbon bonds. And in a carbonyl, there is also a double bond to oxygen. And a carbonyl is a polar functional group. A carboxyl is attached to a carbon and double bonded to an oxygen and single bond to a hydroxyl on this end. This hydrogen is easily lost from this functional group, therefore it is considered acidic. So the properties for this carboxyl functional group are acidic. And another functional group is an amino functional group. This has a nitrogen in the middle, and it is bonded to a carbon, a hydrogen, and a hydrogen. And this can gain a hydrogen molecule, therefore it is considered basic. And then we have our phosphate functional groups, which are a phosphorus double bonded to an oxygen and then single bonded to 3 more oxygens. This is another functional group that can easily lose a proton, therefore it is considered acidic. Here is another table I found online, and what I'm doing with showing you multiple tables is showing how these different functional groups are represented in ball and stick models. So you have, again, this R, which is a carbon attached to an oxygen and a hydrogen. So in the previous slide, it didn't show, it showed the oxygen and then a bond and then a hydrogen, but here it's just showing the carbon attached to an OH. We have another column in this table talking about the properties. So this says it's polar, but it also talks about how it's hydrophilic. So it's water loving, if you remember that from the first class. And then we have structures for the carbonyl, the carboxyl, the amino, the phosphate. But in addition to these, it's showing another functional group, which is the thiol. If you watch the video about functional groups in 2 slides before this one, he describes in that video that there are actually 8 functional groups. And in your book, they only show us 6. Sorry. So I will review the 6 functional groups that are in your book. Okay, so what I want you to do is pay close attention to the properties and the examples of these 6 functional groups. The midterm test will ask that you explain the properties and give an example of each of these functional groups. The first functional group that I'll show you today is the amino functional group. The amino functional group has a nitrogen attached to 2 hydrogen and an R group. Glycine is an example of an amino acid that has the amino group attached to one side. It is considered a basic functional group because it can remove protons from solution. The next example of a functional group is a carbonyl. A carbonyl has a carbon within it. It's attached to 2 carbons and double bonded to an oxygen. It is a polar molecule. If you look at the structure shown here in the table, you can see that oxygen has this double bond, so the sharing of electrons, a lot of the electrons are going toward the oxygen, so this is going to have that negative charge on this side, and these carbon bonds are going to be more positively charged. An important biological molecule is acetaldehyde. You can see this carbon is here, the double bond to the oxygen is here. You have another, we have a carbon-carbon bond shown here, and then the hydrogen group is here. And again, for the test, I'll give you the structure with a carbon double bonded to an oxygen. You'll have to recognize that with this double bond, the sharing of electrons is more favored on the oxygen side of the molecule, therefore it will have a polar nature just like water. And that acetaldehyde is an example of a biological compound that has a carbonyl functional group. Carboxyl is another functional group that I want you to know, and it has a carbon double bonded to an oxygen. There's a single bond to an oxygen and then another single bond to an oxygen. So the property of a carboxyl is not that it's polar, but instead it's considered acidic. So if we look at acetic acid, which is an important biological compound, we see this hydrogen hanging out, kind of dangling off on this structure. That hydrogen is considered easily reactive to within water, so it'll lose this proton and increase the acidity of whatever water or whatever solution it's in, so therefore a carboxyl functional group is considered acidic. Phosphate is the functional group that has a phosphorus attached to oxygens, attached double bonded to another oxygen. Oh, sorry, it's double bonded to 2 oxygens and single bonded to 3 oxygens. It also is considered acidic because it can lose a proton to whatever solution that it's reactive with. The biological molecule that is that has this phosphate group is this 3-phosphoglyceric acid. The hydroxyl is a functional group that has an oxygen attached to a carbon group and also attached to a hydrogen. It is polar, much like the carbonyl, so there is a sharing of electrons toward an oxygen molecule that is not equally, the electrons aren't equally shared to the carbon or the hydrogen. So we see this hydroxyl group hanging off here on ethanol. Here's the R group. You see R here? That's what this is here, this these 2 carbon molecules with the hydrogens coming off. And your hydroxyl is this OH at the end. Methyl is a functional group that has 3 hydrogens and a carbon attached to an R group. This is shown when the example isoleucine, an amino acid that has a methyl group shown here with a red box. Another example is cholesterol. It has methyl groups attached to it as well. The methyl functional group is nonpolar. The next section of the course is protein structure and function. To begin talking about protein structure and function, we must first talk about the early origin of life experiments. I'm talking about Miller's spark discharge experiment. You can see the lab for this week for further explanation and design, but based on the Miller's spark discharge experiment, we now know that we are able to produce biological compounds without life, only with heat and electrical current and gases. The products of the Miller spark discharge experiment that were of interest were the production of formaldehyde, hydrogen cyanide, and amino acids. Amino acids are organic compounds that contain both an amino and a carboxylic acid functional group. There are three categories of amino acids. There are the nonpolar side chains, the polar side chains, and the electrically charged side chains. I have here an example of a table that shows 20 amino acids found in organisms. There's an example of this table in your textbook. These tables will divide the amino acids up into the characteristics that make their R groups different. Every amino acid is a carbon atom attached to an amino group, and a hydrogen atom and a carboxyl. So the middle here is carbon, and then everyone you see has a nitrogen with 3 hydrogens attached to it, and everyone has this carboxyl attached to it. What's different is what is highlighted in blue in this table. The glycine has a hydrogen R group. The alanine has a methyl R group, and so on and so on. And what I want everyone to pay attention to is the description of these R groups. These R groups are nonpolar and aliphatic, whereas these R groups are polar, uncharged R groups. And then there's the positively charged R groups and the negatively charged R groups. And then the non-polar aromatic R groups shown here. So now that we've covered the 20 different types of amino acids, I want to start us talking about how amino acids polymerize to form proteins. So amino acids are also called monomers. So if we look at this figure on the right, we see that sugar is considered a monomer, and when they polymerize, they form a polymer called a starch, which is just a series of this sugar molecule on repeat. So sugar and a sugar attached to a sugar. Now, if we think of an amino acid as a monomer, then if we have a long chain of amino acids, then we call that a protein. And the same goes with nucleic acid. So the deoxynucleic acid is the monomer, and then the ribonucleic acid is the polymer. When we if a monomer polymerizes through condensation, so that's the generation of water, they form macromolecules or proteins. So this is a reaction from your book where they show a carboxylic acid reacting with a amine. If you add heat to this reaction, you generate an amide and a water molecule. This is what is called considered a condensation reaction. Just to zoom in on this concept of how peptide bonds are formed to form polypeptides, here is a nice image of two amino acids. This is amino acid 1, and you have the carboxyl group shown here. You have the hydroxyl, the O and the H highlighted here, and then here is the amino group of amino acid 2, and you have the hydrogen atom here highlighted. We see that when these two react, depicted by this green arrow, that the hydroxyl is removed, the hydrogen is removed, and you get water. And this strong peptide bond is generated between the carboxyl group of amino acid 1 and the amino group of amino acid 2. So, therefore, at this point, we should know that amino acids are small molecules that have unique R groups. Polypeptides or polymers are made up of amino acids. When the carboxyl group of one amino acid reacts with the amino group of another amino acid, a strong divalent bond called a peptide bond forms. Small polypeptides are called oligopeptides, and large polypeptides are called proteins. Proteins are molecules that are responsible for catalysis, defense, movement, signaling, structure, and transport in cells. The book has several examples of what proteins look like. So keep in mind that these are long chains of monomers or amino acids that are linked together and folding upon themselves. In this slide, I have on top a structural protein called tubulin, an enzymatic protein called DNA polymerase. This is the enzyme that helps us in the lab at the SSRC do reactions called polymerase chain reaction. We have regulatory proteins. This is an SRC kinase. And we have carrier proteins such as hemoglobin. Again, this is just an example. These are examples of what proteins are good for, and these are examples of what proteins look like. To end lecture today, I will have you guys watch this short video about the diverse sizes and shapes observed in proteins.